Here we are at part 2 of the blood. In this lecture, we're going to talk a little bit about white blood cells and describe each one and some of their roles. And we will also then jump into talking about hemostasis, which is how the body will stop its blood loss through clotting, the clotting response. And then we'll talk a lot about blood typing, which is kind of an important clinical topic for probably most of you to consider. All right, first up, the leukocytes, or the white blood cells. So a couple characteristics of all white blood cells that make them a little bit different than erythrocytes is that there are several different kinds of white blood cells. When we're talking about red blood cells, there's really just the red blood cells, but the white blood cells, there's a few different types. They're briefly summarized below. We'll talk more in detail about them here in another slide or two. But I put this slide up just to introduce you to the fact that there's 5 of them, and the order of abundance in which they are found in the blood is best kind of summarized with a mnemonic, and there's a lot of different mnemonics out there, but the big popular one is never let monkeys eat bananas. Now, just to be clear, I think that monkeys should eat bananas, but in this in this mnemonic, this is how it shakes out. The letters of these words will tell you the relative abundance of these leukocytes. So the most abundant in your body are the neutrophils. The second most abundant are the Ls, or the lymphocytes. The third most abundant are the monocytes. The fourth most abundant are the eosinophils. And then the most rare or least abundant would be the bananas, or the basophils. Okay, so that's a clever little mnemonic that you can use to remember just about how abundant these are. What's interesting about white blood cells is that they travel in the blood for a very short time. It could be as short as a couple of hours, or it could be, maybe on the order of a couple of days, or even maybe some weeks, but it's certainly not as long as a red blood cell, which travels in the bloodstream for, on average, 120 days. These white blood cells will be made in response generally to some need or demand in the body. They'll go do their job, and then they will retire. They won't be in the blood for very long. What's also very interesting about these is that they have an extra kind of like superpower of being able to travel throughout the body. Red blood cells are just like big sacks of hemoglobin that, cruise around in the bloodstream. But the white blood cells are mobile. They can get into and out of the red blood of the bloodstream, out of the blood vessels, and they can find their way into different nooks and crannies of your body. So that's really fascinating because think about just a little blob, a cell, being able, a cell doesn't have feet or little hair cells or like flippers or fins, and it's still able to move around in your body through a pretty extraordinary mechanism. That mechanism is called diapedesis, and we'll look here in a minute to describe what that how that looks. And then the way that they find where they're supposed to go in the body is called chemotaxis. Now, what these look like is, imagine there is some sort of infection or wound someplace in the body that needs healing. Okay, so in this picture we're seeing you can imagine the stratified squamous epithelium of the epidermis, and let's say there was a cut and some bacteria got into that cut. Well, as those bacteria are coming in contact with, there's a couple different cells in here that would recognize them, or if these are being destroyed somewhere, they'll release a whole host of chemicals, which generally we sometimes refer to as cytokines. Cytokines is a broad name describing any type of chemical that elicits an immune response. But in either case, there'll be a whole bunch of these chemicals that are released, and they will make their way into the bloodstream. And white blood cells can detect those chemicals that are released, and they will continue to move towards the site that has the greatest concentration of those. So here, there might be a lot of those chemicals. It's a highly concentrated place. There's a high concentration of those chemicals here, and maybe it's a little bit more dilute. The chemicals have spread out, and so they are actually able to detect kind of the concentration gradient and move up that gradient to the site that's most has the highest density of those cytokines being released, and that's likely going to be the place where the white blood cells can start to join the fight of the infection. Now, when these cells do detect that there are the cytokines being released, it will activate a whole cascade of events which will cause these cells to actually stick to the walls. There's a whole bunch of, and I won't go into all of this detail now, but it's quite complex. There's a whole bunch of little receptors that get inserted into the endothelial cells of the blood vessel, and those receptors will bind to the white blood cells. Now keep in mind the white blood cells are probably cruising by really fast in the bloodstream, and so they have kind of this first thing that happens where they attach to the endothelial cells and they start doing what's called a slow roll. They kind of roll, and then finally they anchor themselves in, and then they're actually able to wiggle their way through the junctions here, the endothelial cells. Endothelial cells are, for the most part, pretty tightly bound together, and these cells are able to bend their shape, squish themselves in between the endothelial cells. How they do that is a total mystery to me. How does a cell know how to fold itself in half and squish its way through two other cells? I don't know. But it's a pretty remarkable process known as diapedesis. Diapedesis is the actual migration of the cell out of the bloodstream and into now the tissues. And then from the tissues, it can kind of get wherever it needs to. So in that sense, white blood cells have a very cool property. Here's a picture showing a white blood cell squeezing itself through the endothelial lining. And then once it gets to the site of infection, it can start waging war against the pathogens that have infiltrated the body. There's a cool little video here that will show kind of an animation of this if you're interested to see this process. And it doesn't have any sound, but I'll kind of narrate it for you. One of the biggest cytokines is this TNF-alpha, which stands for tumor necrosis factor alpha. That will be released. It will diffuse. Here what you're seeing is the little spindle-shaped smooth muscle cells that surround all of your blood vessels. Okay, so and then you can see the extracellular matrix that has maybe some collagen and fibers and things sticking out of it. And then we're going to descend here in a second into the inside of the blood vessel. And now we see the endothelial cells, and here's all of the white and the red blood cells. They've removed the red blood cells for us, and now they're just showing us the white blood cells so we can see them a little bit more clearly. And they're, as you can see, flowing pretty fast through these blood vessels in your body. But those cytokines that are released will cause the endothelial cells to secrete kind of a receptor that is sticky to the white blood cells. And that will the white blood cells will kind of roll, and then they'll anchor themselves in. They'll come to a halt, and that's called the stable adhesion. And then once they've come to a halt, then they begin their process of sneaking through the endothelial cells, also the smooth muscle cells, and the extracellular matrix, and then voila, now they're out of the bloodstream and anywhere in the body, in the tissues. Okay, let's look a little bit more in depth at the different types of white blood cells and kind of their unique roles in the body. You can divide all of those 5 leukocytes into 2 classes. 3 of them are what we call granular because they have little granules of chemicals inside of them, and the other 2 are what we call agranular. The granular leukocytes are, 1, the neutrophils. These are the most abundant. About 50 to 70% of all of the white blood cells are neutrophils. And I like to think of these as being kind of like the Marine Corps of all of the soldiers of the immune system, in the same way that the Marine Corps of the United States Army or United States military is well known for being highly mobile. They can get to any place in the world within hours. So neutrophils are highly mobile, and generally these are the first ones to any big immune response. They're highly capable and well suited for diapedesis and migrating through the body, and also because they're the most abundant, it's also the highest probability that they'll be the first ones to show up on scene, simply just because there's so many more of them. They are able to phagocytize pathogens. That means basically consume or eat pathogens. They're also cytotoxic. Cytotoxic means they have chemicals in them that can kill other cells. In their granules, they have these compounds called defensins, which are basically a type of chemical that can poke holes into the membranes of pathogens. And once you poke holes into the membrane of pathogen, it destroys the kind of the selective permeability of the cell, and no longer the cell can regulate what's in and out of it, so then it dies. It also has lysozyme inside of the vesicles inside of its cell. So lysozyme is a kind of an enzyme that breaks down substances. And it also has things that are kind of comparable to like bleach or hydrogen peroxide that are able to oxidize things. Oxidation usually means to break molecules apart from polymers down to monomers. If you note the shape of these, they have kind of a multi-lobed nucleus. Sometimes this is called a polymorphonuclear leukocyte because it has these little bulges on it, but that's kind of above and beyond what you need to know. But it does help to kind of recognize this by looking at the multiple lobes. Okay, next up are the eosinophils. Our characteristic feature is that they're more red staining. You can compare the cytoplasm of the neutrophil to the cytoplasm of the eosinophil. You'll see it's a little bit more red in color. Also, if these have a multi-lobe nucleus, but generally it's only 2 lobes. And oftentimes it will look kind of like this. It looks like there's 2 lobes connected by a thin branch. Sometimes the branch here will be thin, other times it will be more thick, so then it just kind of looks a little bit like a C shape. But it has kind of that general shape. Some people think this looks like an old school telephone receiver. They're a little bit more rare in the body. Only 2 to 4% of them, of the white blood cells, are eosinophils, and they help to reduce inflammation. So inflammation is a response that your body will take to help increase blood flow to a certain area, and sometimes it's a good response, and at other times it's a bad response. Your body needs to be able to kind of turn inflammation on and then also turn it off. If it couldn't regulate its inflammatory responses, it would lead to a lot of kind of poor function in parts of the body. So these are kind of like the off switch. They help to reduce inflammation when inflammation has run its course and is no longer needed. What's interesting about these is we generally find them increasing in the blood if there is an allergic response. Also, they're usually going to be elevated in the blood when there is a parasitic invasion in the body. And I will say that you might be thinking, parasites, I mean, aren't those really rare? But there's actually, I would probably venture a guess that maybe even you have some very small little parasitic creatures going around in your body somewhere. They're probably a little bit more common than you think. There are some really drastic parasites that would make you really, really sick. Some examples are the following, and full disclaimer here, if you're squeamish, don't look at the next like 30 seconds of this lecture. Tapeworm could be a type of parasite that is in your body. These are actually quite large. As you can see, this is several inches long. These are parasites that find their way in the intestines, and they kind of anchor into the lining of the intestine and just hang out there and get a free meal by absorbing the nutrients that are passing along the digestive tract. These are blood flukes. There's a whole class of flukes which are kind of like a flatworm, and these are a little bit more microscopic. There's other flukes like liver flukes is another type. But these are, an infection of these would lead to an illness which is known as schistosomiasis. Or there could be something like a roundworm, and there is a whole bunch of roundworms. Some are big, some are microscopic. If you ever really want to see something interesting, don't do it if you're squeamish. Just type in roundworm into Google Images and see what you find. It will be kind of a scary thing. But there's a lot of different parasites out there. Any infection of parasites would lead to an increase in eosinophils. Again, those examples I've just shown you are more extreme examples, but there are probably more subtle microscopic parasites that are more common that require the defense of eosinophils. They'll basically, what would happen is if there was a kind of a small parasite, the eosinophils would surround it and basically eject their the chemicals in their granules out, and those chemicals would work together to destroy the parasite. These are also phagocytic, so they can consume bacteria or things like that. The third granular leukocyte is called a basophil. This is the most rare, less than 1%. And the nucleus of this one is usually not even really visible, especially on a microscope slide, because the granules here are so dark staining. They're very, very dark purple. These are interesting because these actually kind of encourage inflammation in that they release a substance called histamine. So these kind of would increase inflammation, whereas these would kind of decrease, work to decrease inflammation. So you get a little bit of a regulation between the two there. The next would be the agranular leukocytes, and these are going to be the monocytes. Monocytes are the largest. They're fairly abundant in the body, and these are really good at engulfing things. So they kind of will phagocytize pathogens, but also even debris. If, let's say, there was a cut and maybe some dust or some like a wood splinter or foreign material was entering the body, these would work to kind of start consuming that, and they do a good job because they're of their size. These are interesting because there's a couple different forms. A monocyte refers to the name of this cell when it's in the blood, but oftentimes these won't just stay in the blood. They'll enter a tissue, and as soon as they leave out of the bloodstream and now enter the tissues of the body, they're now called a macrophage. So maybe you've heard of that. Macrophages can be either free or fixed, meaning they can either stay in a permanent spot or they can move around in the tissues of the body. The characteristic shape of a monocyte is kind of to have a kidney bean shape like this, is what people often describe it as. Sometimes it's really kind of like C-shaped like this. Other times it's more just kind of like barely a kidney shape. But these are easily recognized by their, I think, easily distinguished by their size in comparison to the other white blood cells. And then we have a whole bunch of other white blood cells, which are all collectively lymphocytes. These are going to be the second most common white blood cell found in the blood. They are abundant in lymphatic tissues, so these would be like lymph nodes, or there's what are called mucosal associated lymphoid tissues, or MALT. Those are just kind of regions where there's clusters of lymphocytes found throughout the body. Usually they're in the digestive tract. And they have different roles. So we have what's interesting about lymphocytes is that there's several different kinds. There's what are called NK cells, there's T cells, and there's B cells. Again, we'll learn more about these when we get to the immune system chapter, but for now, kind of what we generally would summarize these as their functions is the B cells are predominantly well known for their role in secreting antibodies. The T cells are what are well known for being either cytotoxic or what we call helper. There's two different kinds of T cells. The cytotoxic ones kill cells. The helper T cells actually help the B cells make antibodies. And then the NK cells, these are going to be involved in what's called immune surveillance. And that's basically checking the body for abnormal cells, and they can actually destroy mutated abnormal cells, such as those that would be involved in forming a cancerous tumor. All right, a little bit of practice here. You know, it's one thing to see these on a nice cartoon. It's another thing to see these on a actual slide of blood. So I'm just going to put up this slide here for you. This is kind of a cool slide because every leukocyte as well as a platelet is on this image. So go ahead and pause the video and see if you can guess which one is which. Okay, the answers are the following. We have over here the big cell. You know, you can sometimes use you can sometimes use the red blood cells, that's these things, as almost measuring sticks to figure out how big these are. A monocyte I usually think of as about 3 red blood cells wide, whereas the rest of these are more like 2 red blood cells wide. It's kind of a crude way of measuring, but that's kind of what I do. You can also see the little characteristic kidney bean shape here. The neutrophil you can see is the multi-lobed one, and again these are the most abundant. Here's our eosinophil. Note the color between these of the cytoplasm. Because of the granules that are in here, it stains darker red, and this is more of a pink or light kind of bluish color. So that's one way to tell the difference here. We also see our 2 lobes connected by our thin branch, so that's also kind of a tell. And then here's the basophil with its very dark purple or dark blue staining granules, and usually when you look at the edge of this, it's gonna kind of look like this, where you can it almost looks like it's got these granules popping off the surface. It's not like a smooth surface around the outside. Now, one that most people get confused is the lymphocyte in the monocyte or the lymphocyte in the basophil. The lymphocyte oftentimes will be totally just dark staining, because for whatever reason most of the cell is going to be this this dark body here. This one is actually showing us a lot of cytoplasm around the perimeter, which not all of them look like that. Sometimes the entire cell is dark and there might only be just a tiny little sliver of cytoplasm visible. And then this one usually appears very dark. You know, you're not always going to be able to see with high resolution each of these little splotches. Sometimes it just looks like a big dark blob. But the big tell is that a lymphocyte is going to be slightly smaller. It's usually you know, less than probably 2 red blood cells wide, and this is usually more than 2 red blood cells wide. It's actually a pretty big cell. And then also, you'll always, when you look at a lymphocyte, you should always see a little bit of a sliver of a clear cytoplasm. Even if it's small, it should still be there. You won't see that on the basophil. There's going to be dark purple splotches around the whole cell. There'll never be like a region where it's just nothing but kind of a pale color. There's no dark staining parts. There's always going to be the dark staining granules everywhere. And then the other big difference here is between neutrophil and eosinophil, and again, just look at the cytoplasm. On a practical, it would likely be, if there was an eosinophil on there, it would likely be next to another white blood cell, so that you could kind of compare the cytoplasms between them and see that there's something different about the color of this one compared to the cytoplasm of the other one. Here we have our little platelet, and then the rest of these are red blood cells. Couple of disorders here of the white blood cells. The first is what's called leukopenia. This is just when there's a low level of leukocytes. This could be due because there is a disorder of the immune system, like a cancer perhaps, or an infection of one of the immune organs, or it can also be the result of chemotherapy. Chemotherapy is a procedure that will kill any cells that are actively being made, and because white blood cells are being made every day, if they're being destroyed in that production process, it will lead to a low level of white blood cells. Leukocytosis means that there's a high level of leukocytes, so it's just the opposite of leukopenia. And this can be a sign of infection. So if there's an infection in the body's going to ramp up its level of white blood cells to help destroy or win the battle against the invading pathogen. Leukemia is a cancer of the leukocytes, and there's kind of 2 different kinds of leukemias. There's a myeloid leukemia. That would be a cancer of any of the white blood cells that come off of the myeloid lineage. There's also a lymphocytic leukemia, which would be a leukemia of the lymphocytes. In either case, usually it will lead to an abundance, so it would lead to an excess level of leukocytes, so this could lead to leukocytosis. And then not only that, it won't not only would it be a elevation in the number of white blood cells, but also a lot of them would perhaps begin breaking down. They wouldn't be functioning as they should. So there'd be a lot more of them, but perhaps they're not actually working anymore or doing what they're supposed to be doing. Then lastly, we have a lymphoma. Lymphoma is going to be a malignant cancer of the T or B lymphocytes that accumulate specifically in the lymph nodes. So this is different than, let's say, a lymphocytic leukemia. Lymphocytic leukemia would be a leukemia in which there's a cancer invading the lymphocytes that are in the blood, but this would be a lymphoma is a cancer of the lymphocytes that are not in the blood but anchored somewhere in one of the lymph nodes. All right, this is a good point to maybe stop and do a little review. So see if you can list and describe the features and roles of all of the 5 leukocytes that I've mentioned, and then also describe the 4 disorders that we had just presented. The next part of this lecture is all about platelets and the clotting response. This is given the name hemostasis, which literally means stopping the flow of blood, basically. Stasis means to stop or have something stay still, and hemo means blood. Platelets, by way of review, remember this is kind of our myeloid lineage, and here's all the cells that are made out of the myeloid lineage. One of them is called a megakaryoblast, which turns into this really interesting, huge, colossal cell called a megakaryocyte. And megakaryocytes have these little cytoplasmic extensions that kind of bleb off of them. And what will happen is, let's say this is a blood vessel. Those cytoplasmic extensions will kind of find their way into the bloodstream, and then the flow of blood actually cleaves them off, and then they form platelets. Now the body can regulate how many platelets are made. One of the ways it does this is it releases a hormone called thrombopoietin, and thrombopoietin will drive the production of megakaryocytes, and the more megakaryocytes there are, the more platelets will be made. Platelets are not actually cells because they're just kind of cytoplasmic contents of another cell, but they still have mitochondria in them, so they can make energy. They have microtubules in them, so they can kind of have a shape or structure. And they also have granules inside of them that are going to have a bunch of different signaling molecules, most of which will have the function of vasoconstriction, which is good because usually these are involved in stopping blood flow or blood loss. So vasoconstriction is a helpful way for the blood vessel to do that, because it will kind of shut down and squeeze itself so that there's not as much of an opening for blood to escape. Now, there are 3 phases to hemostasis, the clotting response. The first is what's called vascular spasm. The second is what's called platelet plug formation, and the third coagulation. And this is just literally what we mean when we say clotting. These look like the following. So first, there will be an injury. All right, so this is showing that a blood vessel, a small blood vessel, has been severed. And shortly after the severing, within about 30 seconds, there's going to be vascular spasm. So what that means is that the smooth muscle that's lining this blood vessel, the smooth muscle kind of can wrap around in like a ring type structure. And when those smooth muscle constrict, it will actually pinch down the vessel. So you're seeing the vessel's actually been pinched down. That's called vascular spasm. And it happens really fast, and it's going to greatly reduce the amount of blood flow passing through there. Okay, then the next step we said is the platelet plug formation. This is kind of interesting. Platelets are actually activated. They are activated by 2 different things. One, it could be that there's some collagen from the extracellular matrix of this tissue that's sticking out, and when the platelets detect that collagen, they bind to it and become kind of activated. Platelets have these little things called pseudopods, which are just like little kind of extensions that you can see them here. These little pieces that stick off of them, and those are really good at grabbing on to something like collagen, which is going to be kind of sticking out if this vessel is cut. Also, they can be activated by just chemicals, cytokines released by damaged cells in this area. So either of those two ways, the collagen being stuck out there or the chemicals, will lead to kind of an increase in how sticky they are. And they're really good at grabbing onto collagen. There shouldn't be collagen really exposed in the inner lining of the blood vessel normally, but because it's cut now, some of the collagen that was in these connective tissues is exposed, so they grab onto it really tight. Then usually the platelets will, what we would say, degranulate, and that means they'll release the granules that are inside of their cytoplasm, and some of the things that they'll release is serotonin. Serotonin is going to be a vasoconstrictor, so it will, vasoconstrictor, so it will actually further close down this vessel even above and beyond where it already has gotten due to the vascular spasm. They'll also release ADP, and ADP, adenosine diphosphate, is going to be something that is going to actually lead to attracting and degranulating other platelets. So it's kind of like this domino effect where one platelet sticks and starts releasing its things, and it leads to the activation and degranulation of all of the other platelets that are coming by. And then there's another substance called thromboxane A2, which is going to actually promote the stickiness of the platelets clumping together and further vasoconstriction. So in essence, then basically we're forming the main idea. If we don't remember any of this stuff, the main idea is that we're plugging this gap full of these sticky little platelets. Now the platelets are going to be sticky and they're kind of gooey and they can clump together, but they're not super structural. They're a quick kind of sticky patch is a way to think of it. So in order to make it a little bit stronger and to provide a kind of a scaffold on which tissue repair can start to occur, we need to get to coagulation. In coagulation, the big thing that happens here is the production of this structural fiber, which is called fibrin. The fibrin will insert its way into here, and if I kind of zoom in so you can see this a little bit better, you can see that the fibrin is illustrated with these like little stringy yellowy things. And here you can see kind of an interesting scanning electron micrograph of the fibrin and the red blood cells and the platelets all kind of sticking together here. Now, how fibrin is made is a very, very, very complicated process, which I won't ask you to know, but I do want you to know at least some of the process. It looks something like this. There is dissolved in the blood a substance which is called prothrombin. And there's a factor which is called factor 10. And when factor 10 comes onto the scene, it turns this thing which is called prothrombin into an activated form which is called thrombin. Anytime you are looking at biochemistry and you see a word that says pro, that means that it is something that is not active but can be turned on to then go do something. So prothrombin is inactive, it gets turned on, so now that's the active form, which is just called thrombin. Thrombin, the word thrombin refers to anything dealing with a blood clot. Thrombin then will turn on this other thing in the blood, which is called fibrinogen, and fibrinogen, once it's activated, it gets converted into fibrin. And fibrin is first made with monomers, and then with this additional factor, factor 8, those monomers all link up to form the network of strands. But in either case, it's pretty cool because this is dissolved in the blood, and these are all soluble proteins, and then this is actually insoluble. So this kind of structural protein just comes out of nowhere, in essence, precipitating out of the fluid plasma. Now, if you think that was complex, take a look at this. We said factor X is what kind of gets this part started, which is called the final common pathway, but how we get to factor X is even much more complicated. That's this pathway here. All right, so you have factor 7 getting converted and turning on, and then that turns on factor 11, and then factor 11 turns on factor 9, and then factor 9 is going to get converted via factor 8 to form this complex, and that turns the inactive form of factor X into the active form of factor X, and then that, with the help of factor 5, will then take prothrombin and turn it into thrombin. Okay, I'm not going to require that you memorize all of this. We'll save that for medical school if you ever go. But I do want you to at least know this kind of final common pathway of how fibrin is made. Prothrombin to thrombin, that turns fibrinogen into fibrin. Okay, can you do that? Can you handle that? I hope so. Also, just a difference here between what's the intrinsic pathway and the extrinsic pathway. Intrinsic pathway would be damage to the actual vessel wall, so the blood vessel wall, or even some of the blood vessels themselves. The extrinsic pathway would be damage to the cells that are surrounding or around the blood vessel. So that's kind of the big difference there. Okay, so that's how blood clots are formed. Now, one last bit here. At some point, the blood clot has to go away. The body has to get rid of it. And this is a process known as fibrinolysis. This word literally means the breaking apart of fibrin. And how this works is essentially there's a protein which is called plasmin. Plasmin is made from a substance called plasminogen, same as kind of fibrinogen. Plasminogen, which is inactive, gets converted to the active form known as plasmin, and then plasmin, this conversion here is also made by another protein, which I want to ask you to remember. But plasmin will then go in, and then it will dissolve, basically, or break down fibrin. And once fibrin is broken down, then the platelets and everything are kind of freed up, and the body can start to rid itself of the clot and hopefully replace it with repaired tissue. Okay, a couple of clotting disorders. Hemophilia is one. Hemophilia is basically a genetic disorder in which there's a mutation that leads to a clotting factor that's inactive. And I don't know, it probably varies, you know, that would mean if we're looking back here, it's a mutation that makes it so that one of these doesn't quite do its function as it should. And I don't know specifically which one, and there's probably a lot of different types of hemophilia which involve different types of clotting factors, but you can also, you know, if you can imagine, if just, you know, one of these is missing, then this whole cascade could be affected. And people with hemophilia have a hard time forming blood clots, so if they even cut themselves with a razor, and it's a smaller cut, it will bleed and bleed and bleed and bleed and bleed, and it's a kind of a problematic disorder. They usually, if you read a textbook about hemophilia, they'll give an example about the royal family of England. There was, I think it was England, there was a lot of kind of ancestral kind of inbreeding in those families at that time, and so if there was a mutation, such as the one that leads to hemophilia, it kind of stayed in a higher concentration in the gene pool, so there was more of an incidence of hemophilia in those families. I also like to mention here, kind of how interesting it is. You might ask, why is this so complicated? Why does the body have to make it this way? Why can't the body just, you know, make fibrin? And I'll say that oftentimes you see that very severe responses that can be problematic if accidentally turned on usually have a lot of steps like this to prevent them from falsely firing. If there was a blood clot formed when there wasn't supposed to be a blood clot formed, that's a very bad thing. Okay, and so that's why so many steps are required, because if it was really easy for this process to happen, it might lead to more higher incidence of false alarms in blood clots being made when they're not supposed to. So that gets me to the next disorder that we're going to talk about, which is known as a thrombus. A thrombus is a blood clot that blocks a blood vessel. So it blocks blood flow. And this is a great thing if we're talking about a severed vessel which has been cut. It's a very bad thing if we're talking about a normal blood vessel that's supplying blood to an organ. Specifically, if a blood clot does block a blood vessel, we call it a stroke. And we call it a stroke if it is to the brain, and we call it a heart attack if it's to the heart tissue. So the heart and the brain are kind of two important organs of particular importance that we usually talk about when we're talking about a thrombus. Now, if that thrombus, which is a blood clot, actually breaks off of the blood vessel and now is traveling throughout the body, it's then called an embolus. And an embolus is particularly scary because if a blood clot is going around in the bloodstream, once it goes down a few branches into a smaller vessel, it can actually lodge into that vessel and cut off blood flow to a lot of the different tissues that are downstream of that blood flow. A place where this happens probably more frequently is in the lungs, and if that's the case, it's called a pulmonary embolism, or pulmonary embolus. And if it's large enough, that can actually be fatal. So a little bit scary here, and that's why you can see how important it is that the body has such a complex process for making a blood clot, because if they're made in the wrong time or the wrong place, it could be potentially a fatal event. All right, take a little time to review here. List the 3 phases of hemostasis, see if you can recall them, and then a little description of each. Tell me a little bit about how fibrin is produced. Tell me a little bit about the difference between the intrinsic and extrinsic pathway. Don't try and draw out the whole intrinsic or extrinsic pathway, just a sentence or two about the difference between these, and then what is fibrinolysis, and also identify the most important protein in this process. Sorry, this is kind of redundant here. This is the question. And then list and describe 2 clotting disorders that I have just presented. All right, our final part of this lecture is to talk about blood typing. And I will say on the surface that learning about blood typing is very, very simple. And then in about 30 seconds, it gets very, very complex. So I'll try and guide you through it as clear as possible. But on the simplest side, I think you'll all be able to get this with relative ease. The way that we name one's blood type depends on what type of antigen we would call is on the red blood cell. Antigen, this word here, literally it's a general word that describes any type of recognizable substance that's attached on the surface of a cell. So for example, here is a picture of a red blood cell. That red blood cell has all of these little identifiable markers on it, which we would call antigens. And if we're saying that this is type A blood, it's called type A blood because the particular antigens on here are what we would say A antigens. So if a red blood cell has A antigens on it, we would say that that is type A blood. Okay, simple, easy peasy. Similarly, if a red blood cell has what we would call B antigens on it, these ones, then we would call that type B blood. Okay. If a red blood cell has both A and B antigens on it, well, then it's type AB blood. And if that red blood cell has neither A nor B antigens on it, then we would say that it's not type A or B, but it's type O. So type O means there are no antigens. So this is all very simple and pretty intuitive. Now, what gets tricky about the ABO blood group, and I'll also point out too that there's a lot more antigens above AB, but these are the most common, so this is usually the only ones that we talk about. Now, where this gets tricky is when we think about the antibodies that are in the plasma of individuals with these blood types. Generally, you have in your blood the possibility of making antibodies towards anything that is not supposed to be in your body. We'll learn more about it in the immune system chapter, but your body has a really great ability of making antibodies to anything that's foreign, and that helps you destroy anything that's foreign in your body. you should not have any antibodies that bind specifically to things that should be in your body, because then that will target things that are in your body for destruction. And that would be bad. That's called an autoimmune disorder when that happens. Okay, so somebody that has type A blood, they do not have a B antigen on their red blood cells. So the B antigen is foreign to them, and so they'll likely have anti-B antibodies in their plasma. The way that we name an antibody is we call it anti, and then whatever it actually specifically binds to. Antibodies, one of their characteristic features is that they will specifically attach to things. So they're able to recognize and identify specific things in the body. So an anti-B antibody binds specifically to these antigens and these antigens alone, which is a good thing because if foreign blood gets put into this person's body, their anti-B antibodies will target it for destruction because that blood's not supposed to be in their body; it's foreign. Similarly, somebody with type B blood, they do not have the A antigen, they have the B antigen, so they would have the anti-A antibody, which would actually recognize and destroy anything with the A antigen. Remember, we name these anti and then whatever they bind to. AB blood, see if you can guess what type of antibodies they would have. We know that they could not have the anti-A antibody because if they did, it would bind the A antigen and it would destroy their own red blood cells. We know they also cannot have the anti-B antibody because that would bind to the B antigen and destroy their red blood cells. So folks with type AB blood, they actually don't have either the anti-A or the anti-B antibodies in their plasma. And then lastly, type O blood, they have both anti-A and anti-B antibodies because they don't have any of those antigens on their red blood cell, so they have those antibodies in their plasma. Now, when you're considering kind of the compatibility of blood transfusions, like who could receive blood from who and to whom could you give blood, you have to keep in mind the antibodies that are in the plasma. And I will say that generally the way that it's done is that if somebody was going to donate their blood and that blood was going to be given to somebody, they would take that blood, which is called whole blood, they would spin it out and separate the red blood cells from the plasma, and then they would give that individual just the red blood cells, or what we would call the packed cells. So keeping this in mind, let's kind of think through this. Let's say that somebody with type A blood needed some blood. Okay, they have in their plasma anti-B antibodies, so we would know for sure that we can't give this person anything that's got a B antigen on it, because lurking in their plasma is the anti-B antibody ready to destroy anything with a B antigen. So we could not give this person B blood. We could not give this person AB blood, but because they both have the B antigen. But we could give this person O blood. Okay. Now, people get confused because they go, wait a minute, why can't we give them O blood? Because if we give them O blood, won't we be, bringing along the anti-A antibody, and wouldn't that then come and destroy their red blood cells? And the answer is yes, but remember, we leave these behind. We only take the cells, the packed cells, and give them to the individual, because we're taking this. If type O gave blood, we'd take their blood, we'd spin it down, we'd return their plasma, and we would just take the red blood cells. All right. Okay, we could try another one. Let's say we're wondering to whom could we give blood. Let's say I have type B blood and I want to give my blood to somebody else, and I'm wondering what types of blood I could give my blood to. The answer would be I have to consider, again, the antibodies in the plasma. So if I have a B antigen on my red blood cell, I cannot give my blood to anybody that has anti-B antibodies. So that means type A is out because they've got an anti-B antibody. I could give my blood to type AB if I wanted to, and I could not give my blood to type O because they also have an anti-B antibody. So I could give my blood to anybody that has type B, or I could give my blood to anybody that has type AB. Okay. You kind of want to think through this in two different ways. You want to think for any given blood type, to whom could you give that blood type? But you also, if you were a certain blood type, want to consider from whom could you receive? These are two different questions, and they're going to have different answers. So you have to think about it in two ways. And kind of to illustrate this, I'll talk about these blood types. These blood types are special because AB is what's called the universal recipient, which means they can receive blood from anybody, but it's very difficult for them to give blood to people. That's different than O blood. O blood is the universal donor. So they can give their blood to anybody, but it's hard for them to receive blood. They can really only receive blood from other type O folks. And the reason for that is universal recipient can receive blood from anybody because they don't have any of these antibodies in their plasma. So you could give them B blood, you could give them A blood, you could give them O blood. It's all good. However, the universal donor is very tricky. You cannot give them B blood because they have an anti-B antibody. You cannot give them A blood because they have an anti-A antibody. You certainly cannot give them AB blood because that's a double whammy. The A and the B antibodies would attack both the A and the B antigens. So O blood can only receive blood from O's. But if you're thinking about how they give, if you just take the blood cell and you give it to this person, it's all good. If you give that red blood cell and you give it to this person, it's all good because anti-A antibodies aren't going to bind to anything on the surface. You could also take that red blood cell and give it to type A blood because their anti-B antibodies aren't going to bind to anything on the surface. So it's all good. This is kind of summarized here in the bottom. I think the way that they're writing this is, I think there's type from whom could they receive is the way that they're wording this, I think. So not to whom could they give, because you definitely would not want to give A blood to type O. So if somebody had A blood, they could receive blood from A folks or people with O blood. If somebody had type B blood, they could receive blood from people with B blood or from O blood. If somebody had AB blood, they could receive from A's, B's, AB's, and O's. They're the universal recipient. And if somebody had O blood, they can really only receive it from O. I would encourage you not to try and just memorize these out of your sheer will. If you were to ask me, you know, from whom could these blood types receive, I wouldn't actually be able to tell you, even though I've been teaching this class for many semesters. I wouldn't be able to tell you right off the top of my head. I always have to draw out this chart and see it before I can respond. And so try to not actually just memorize this. Try to memorize this and then use this graph to figure out all of these types of questions. Because if you have this idea in mind with what's happening in the plasma, what's happening on the red blood cells, and then you should be able to kind of think your way through, okay, if somebody had type B blood, to whom could they give? Okay, they can't give it to this person or that person. And I would even invite you on an exam to at least just draw out something similar to these first two rows here somewhere on the exam. I think it would help you out. All right, we're going to keep going here and talk about a special type of blood group called the Rh blood group. So this is not the ABO, this is the Rh. What this refers to is Rh stands for rhesus monkey, which is where the organism which they recognize it. And this is like the plus and the minus story. You know, you not only have type A, B, and AB and O blood, but you also have A minus or A plus blood or B minus and B plus blood. So that's what this blood group refers to. This one's on the surface pretty simple. Basically, if somebody has what we call the Rh antigen on their red blood cell, we would say that blood type is positive. So looking back here, if somebody had the Rh on their blood cell, we would say that that blood is actually Rh positive. If that person did not have the Rh group on there, we would say that that blood type, Oh, sorry, I don't know what I've done. We would say that that blood type is A negative, let's say. All right, so that's this. Now, of all of the Rh antigens, there's one called D. It's a type of Rh. There's lots of different Rh types, but D is the most clinically relevant. About 85% of Americans have it. And what's interesting about the Rh blood group is that the anti-D antibodies are not preformed. What this means is, let's say there's a red blood cell. And let's say we're talking about type A positive blood. Okay, so type A positive blood, they would have the A's, and then they would also have the RHs. Or actually, we could say that these are called the D's. Specific kind of. Now, in this person's plasma, they're going to have anti-B antibodies in their plasma because these are preformed. And the ABO antibodies are kind of weird this way in that they are preformed. The person will have them whether they've been exposed to the B antigen or not. They're just going to have these antibodies in their plasma. But generally, this isn't quite how antibodies work. Usually, the way that antibodies are produced in our bodies is they have to actually see something foreign before they get produced in mass quantities. So a person that has, maybe I should write it this way, a person that does not have the Rh group will not have anti-D antibodies. Okay, so in the person does have anti-B antibodies because they're preformed and bind something foreign. But in this case, the Rh is also foreign to this person, but for whatever reason, these antibodies have to actually be exposed to the antigen before they'll be made. This leads to an important discussion of what we call hemolytic disease of the newborn. This is a problem that occurs if a woman who has a negative blood type is pregnant with a fetus that has a positive blood type and is actually twice pregnant with a fetus with a positive blood type. To understand how this disorder or this disease works, we have to kind of consider the placenta. What you're seeing here, this is a small cartoon of the placenta. Basically, this is the umbilical cord, so this would go to the baby or the fetus. And how it works is that baby's blood will actually come in and kind of go through these, what we call chorionic projections, or just like these little projections of blood vessels that are going to come into very close proximity with mom's blood. Mom's blood, I'll draw it here, is kind of all this stuff here. There's basically a kind of a pool of mom's blood, and through the placenta, the baby's blood can come into very close contact with it. It can get rid of its oxygen, or sorry, it can get rid of its CO2. It can absorb oxygen. It can get rid of its waste. It can absorb nutrients and all that type of stuff. But the blood actually physically never touches one another, which is kind of an interesting component of the placenta. So keep that in mind. Now consider if we were to look really close at one of these projections where the baby's blood vessels will come into close contact with the maternal blood pool. Here we're seeing this is the maternal blood right here, and this is the blood that's going to the baby. Imagine that the erythrocytes on the baby are Rh positive, so that means they have little Rh groups on them. Or, sorry, I keep doing this. We should just call these D, because that's the specific kind. Okay, so they got D antigens on them. Well, what happens is it's no big deal during the pregnancy because there are no anti-D antibodies here. Okay, if there were anti-D antibodies, they could diffuse. Antibodies are very small, so they could diffuse into the fetal blood supply and actually start destroying these red blood cells because they have the D antigen. But on a first pregnancy, that's not the case because remember, D antibodies have to actually be exposed to the antigen before they start getting made in mass quantities. All right. So this is fine because the blood, the fetal blood that has the D antigen is kept separate from the maternal blood that is not having the D antigen. But during the first delivery, when the baby is born, there's a lot of tearing that happens and the placenta is ruptured, and so the fetal blood cells actually get mixed into the maternal blood. This is going to cause the activation of B cells. Those are those lymphocytes that make antibodies, and they are going to start making a whole bunch of anti-Rh antibodies, or anti-D antibodies. This is called Rh sensitization. And now, whereas there used to not be any anti-D antibodies, now, after this immune response, because the mom has come in contact with the D antigen through the fetal blood cell, there's going to be tons and tons and tons and tons and tons and tons and tons of D antibodies made. This is fine during the first pregnancy or the first delivery because the baby's already been born at this point. The baby's already separate from the mom's blood supply. So even though now she has these antibodies being made, it doesn't affect the first born baby. However, it becomes problematic on the second pregnancy because on a second pregnancy, if again the fetus has Rh positive blood, and now mom does have the anti-D antibodies or the anti-Rh antibodies from the from the event that happened on the first exposure. Once the baby is born and the blood supply is made, then mom's antibodies will actually diffuse through the placental wall and into the fetal blood supply and start destroying the fetus erythrocytes, the fetal erythrocytes, and that can lead to the premature termination of the fetus. So that's kind of a problematic thing. All right, and one of the workarounds, which is a pretty clever workaround, is to administer this drug, which is called RhoGAM. And RhoGAM, what it is it's a synthetic anti-Rh antibody, and it would be given right here, right before it would be given to mom right before the first delivery. So there would be these synthetic antibodies that mom would have on hand, and these synthetic antibodies would be there and able to actually grab onto these red blood cells and destroy them as soon as they enter mom's blood supply. Now you might ask, wait a second, how is this drug helpful? It's just destroying fetal red blood cells. But that's a good thing in this case because what it does is it prevents this. If we can destroy these red blood cells, the fetal red blood cells that enter mom's circulation before mom's lymphocytes detect them and start cranking out antibodies, we can avoid this whole immune response. That's a good thing because naturally occurring immune responses such as this one are long-lasting. There will always be these antibodies made if there's an immune response, and that's the case for anything in your immune system. Anytime you come in contact with any foreign pathogen and you have an immune response to it, your body will keep what are known as memory cells around to always have a hand of antibodies available against that thing. But because these are synthetic antibodies administered, they'll do their job and destroy the red blood cells, but then these will be cleared out of the body within several days or weeks, so that by the time we get around to the second pregnancy, they're no longer there. Okay, and we don't have these naturally occurring antibodies still lingering around from the immune response. So it's a little bit of a tricky thing to conceptualize, but I hope that helps to understand. Basically, the bottom line is to prevent. The bottom line is hemolytic disease of the newborn is a problem when a woman is twice pregnant with a fetus that has Rh positive blood and the mom has Rh negative blood. That's when it's a problem. And one of the solutions is to give RhoGAM at the first delivery so that it prevents an immune response that would lead to complications in the second delivery. All right, here's a little review if you would like it for blood type. You could think about, here's the blood types: A minus, A positive, B minus, B positive, AB minus, AB positive, A minus, or O minus, and O positive. Think about what are the antigens that are on the red blood cell. Think about what are the antibodies that are in the serum, and then think about to whom could this individual give their blood, and to whom or from whom could this individual receive their blood. So somebody, for example, that has type A minus, to whom could they give their blood, and from whom could they receive blood? Two different questions, and to do it you have to think about really what the antigens on the red blood cells are and the antibodies in the plasma or the serum. Go ahead and go work through that and see how you do. Again, once you have this whole table filled out, again, I would strongly encourage you not to just try and memorize the entire table. Just learn how to figure out these questions based upon your knowledge and using these two columns here. I wish you the best of luck. Let me know if you have any questions. Another thing that you should also be able to do is kind of explain your way through hemolytic disease of the newborn. I don't think I have a review question on that one, but that's also something you should be able to handle. And this concludes our discussion of the blood. Email with questions if you got them.